We looked at micro debris that was detected in harbor and gray seal samples. And again, we came across those samples while we were looking and recovering hard parts for a diet study. But we were really surprised that we came across some micro debris. So any paper that you read these days on marine micropollution, marine debris pollution, there's two words that are always included in the description, which is pervasive and increasing. And it's a real issue worldwide, both in marine and terrestrial systems. But when we did our literature search, some of the more surprising finds were that it wasn't reported on until 1969. And plastics were developed like in 1907 and then in mass production in the 40s and 50s after World War II. And actually, I read this report recently from National Geographic from the author Parker, and that half of all the plastics ever produced were produced in the last 15 years, which is amazing. So this first paper that came out addressing digested debris was from a paper by Kenyon and Cridler from the Hawaiian archipelago, and they found Laysan albatross had ingested micro debris while foraging off the Hawaiian Islands. And that in turn can kill the adults, but also if the adults survive, they can in turn digest the materials and regurgitate it for the chicks. So it has devastating effects in populations of nesting birds in an area like the Hawaiian archipelago, where you have large patches of debris. Now, the next report didn't come out, and there were two reports in 1972. That's 3 years later. One was from the Sargasso Sea, and scientists finding plastics there while doing plankton tows. But one was much more local, and that was a team of scientists from Woods Hole Oceanographic Institute was actually conducting plankton tows and looking at fishes near a nuclear power plant in Waterford, Connecticut. And they wanted to see how the power plant would impact the local ecology of Niantic Bay. And what they found when they were doing the plankton tows was that they were picking up all these small polystyrene spherules. Polystyrene spherules are basically microbeads. And they were everywhere from northeastern Long Island Sound into Niantic Bay, down to Great Salt Bay in Rhode Island, into Buzzards Bay, and as far south, well, they tested as far south as Vineyard Sound. This is really surprising to me. And they didn't go beyond there, but they believe that quite possibly these small beads could have moved further north and even, up into Canadian waters. So this is a really interesting study. The fish, and this is where it gets really interesting. So when they were sampling fish in Niantic Bay, they found that there were two different types of these beads, polystyrene beads, clear and opaque. And the fish that they collected that had ingested them were targeting the opaque ones, which look a lot like fish eggs. So what they found on the surface of these beads were bacteria and also PC beads that must have been picked up from the ambient seawater. So fast forward 50 years, and here we are doing this study on the diet of seals, but we trip over this material and their scats. So although this project reports on very small pieces, we hope those pieces actually add to the bigger picture for what's happening in the environment. So our intent for the original study was to look at the diet of harbor and gray seals over a period of a year. And CCS was going to be doing the scat analysis through hard part recovery. And we also shared subsamples with Keith Hernandez, who at the time was in graduate school at Louisiana State University. He's now graduated, and actually he presented at the Cape Cod Natural History Conference a couple years ago. He's now graduated. He's Dr. Keith Hernandez, which is really, really great. And he's finishing up the paper on the environmental, the prey DNA that he found in the samples that we processed for the hard parts. So the sampling sites where we worked were South Monomoy Island, Chatham Harbor, Pleasant Bay, Jeremy Point, and also Head of the Meadow. Now, I mentioned the Northwest Atlantic Seal Research Consortium, and I'm mentioning them again because when we began this project, we really thought about how you go about collecting scats. And you actually have to disturb the animals because you visit their haulouts where they rest and recover from traveling or feeding forays. And so what we did was we got as much science as we could when we were in the field. So we would do these surveys of the entire haulout, document all the animals. We would speciate because we wanted to know who we were collecting scats from. And although we would say that typically the seasonal haulouts, and that is the haulouts not during the breeding season, are typically relatively homogeneous in certain times of the year, but you always get these small mixes. And here you got a predominantly gray seal haulout with a harbor seal in the mix. So we tried to speciate, count, and then move in slowly to collect scats. So this is in Pleasant Bay, Chatham area. And the way we move in to collect the scats is we would typically move across the haulout slowly and slowly encourage the seals into the water. Then we'd land and we'd go in to collect the samples. Now, one of the things that was really sobering is how much plastic we used in the process of collecting and storing. So I'm going to introduce Christy in a second. When my collaborator and I looked at what we used, we had to sample all those materials to eliminate them from contaminating our findings. So that changed how we actually went into the field and what we used for collecting. And the way processing scat works is that you have graduated sieves of various sizes. The smallest for our study, which is typical of when we're covering hard parts for dye analysis, is 0.5 millimeters. Now, if you're doing actual work that's targeting micro debris, you want to use even smaller mesh size. But what we do is we would take a scat, we would thaw it, we would clean the exterior of the scat while it's still a little hard to get rid of all the sand. And then you put it at the top of the graduated stack of sieves, and you slowly rinse away the soft material of the scat with warm water. And if you want a little bit of mild soap, you don't want to introduce anything to the scat. And then you're left behind with all these little hard parts. And in these hard parts, we discovered some debris as well. And those hard parts are typically bones or shells or jaws of fishes and cephalopods and shellfish. And just to give you an overview of what you can find, I wanna make sure you understand that the measurements here is in millimeters. So just to give you an idea of what we're looking at, I just looked this up a few days ago. The average size of short grain white rice is 5.2 millimeters. So up here, the cod, the cod otolith, that's about 7 millimeters. And when we use when we actually measure the otoliths or the hard parts that we recover, we use a digital caliper, so we get very accurate measurements. But I just wanted folks to get an idea of what things look like. And the really small stuff like this sand lance otolith, a very common fish in the inshore waters, this area is more closer in size to the debris that we found. And just to give you an example, this is a herring otolith on the right and micro debris on the left that we found. So you can see they're in similar size, and that's why we were able to detect it when we were sieving through the materials. So I have to mention that one of the great things about working at the Center for Coastal Studies is that I work in a great community of people. And my colleague, Christy Hudak, she was trained on a Nicolette spectrometer that's able to identify micro debris. And when we came across the debris in the samples, I was able just to turn to her and say, hey, can you look at these? And she was more than willing to do that. And what we found in the 4 samples that we recovered, I do want to add that we also found a lot of fibers, but there was no way for us to eliminate the fact that much of the clothing we were wearing while we were doing this work probably contributed to the fibers we found, so we didn't include that in our report. But what we did find using the spectrometer was a sample of resin, typical in a lot of marine applications and also construction, a couple of samples of cellophane, and the most complex one that took the most time to identify was the polyethylene propylene diene. And it was the diene, which is a filler for a type of EPDM rubber, which is a construction material like rubber membranes or rubber roofing. I guess that's very common in use in construction. And it was a sample of 2 for harbor seals, which out of 32 scats is 6%, and for gray seals it was just 1%, and that's 2 of 129. But it's important to remember we were not looking for micro debris, we just happened upon it during the processing of the scat. So one of the things I learned from Christy is that we have a library that works in conjunction with the spectrometer that looks at a lot of polymers, but you can get libraries for all kinds of things for looking at fibers, coatings and paints, pharmaceuticals, explosives, and actually one that was really interesting was essential oils. So it's amazing what you can look at with a spectrometer. And if you had questions about that, I encourage you to write to, you can email Christy. She's probably going to kill me, but I know she likes to talk about it, but you could get more information about how she goes about identifying debris samples. So one of the things we had to discuss was, do we believe it's direct ingestion or trophic transfer? That is, the seal had plastics in its system because it actually targeted a prey species that picked up the plastic. So through the prey that it was eating, it was ingesting the plastic. Although, if you think about how many marine mammals interface with their world, with the marine environment, it's often through their mouths. So I have seen, and others have seen, seals actually mouthing all sorts of materials at the surface. So we can't say for certain if this was either direct ingestion or trophic transfer, but based on the type of fish that we saw recovered through the hard parts, many of them are plankton feeders, which basically, like sand lance and herrings of a certain age, will just swim through patches of plankton with their mouths open, ingesting all kinds of materials. And it's very easy for debris to get caught up in large patches of plankton. So we discussed the likelihood of trophic transfer being part of the cause of what we found in the samples. And just to conclude, we had really good matching percentage with the spectrometer and how it handled the micro debris. It was the first report of debris in wild gray and harbor seal population in the North and West Atlantic. But more importantly, it raised a lot of questions, which are, what are the mechanical effects of ingesting plastic? We know that, like in sea turtles and some seabirds and also fishes, it can actually block the intestines, or it can interfere with feeding and feeding mechanisms. There's also the concern about chemical impacts of microdebris, how it may infect the biome, basically the flora and fauna of the stomach and also the intestines. And there's been studies on shellfish and also fishes where it disrupts the immune system. So there's a lot of work to be done concerning microplastics in marine organisms, and there's a lot of work underway. I know of some researchers that are currently working on looking at seal scat and how it might affect the biome. So there'll be interesting reports on the way. And currently at CCS, we're looking at how and if there are micro debris in the pathway of foraging right whales, and we're doing that by collecting zooplankton samples in the area of foraging right whales. And also Christy Hudak is looking at micro debris samples from greater shearwaters and debris found in their gizzards. So there's a lot of interesting projects underway, and very curious to see the outcomes of those. One thing I want to add is that before some micro debris becomes micro, it's macro when it enters the system, and macro debris can be an entangling issue for not only seals, but a lot of different marine wildlife. So we want to encourage people to be careful when they're recreating or near or disposing of trash near beaches, because it can end up on animals. And if you see an entangled seal in the area, call your local stranding organization. And also, when we think about plastics being around for as long as they have, we really have to start thinking about how to block that pathway. How does it get from what we produce and what we use into the system? So although it's a small contribution, it's still important. Think about the what's happened globally, but helping out locally in this beautiful place that we live in. At the Coastal Studies, we Laura Ludwig from the Marine Debris Program and also Jesse Meckling from our education program offer marine debris walks. So please, I encourage you to join them. And also, it's happening all around the Cape. And again, thanks so much for having me speak today, and I look forward to your questions.
Great, thank you, Lisa. All right, we're going to move this right along. If you have questions for Lisa, type them into the chat, as I said, and we'll get to those during the Q&A period a few speakers from now. All right. Yeah, unshare your screen, Lisa, and I will introduce our next speaker. So, moths at the beach. Who knew? Our next speaker is synonymous with moths. There really aren't that many moth experts around, so they're a bit of a rare species themselves. And Mark is the guy. Mark has done rare moth surveys or just general moth surveys for us at Wellfleet Bay Wildlife Sanctuary and just about everybody else, because it's difficult. I consider myself a pretty good all-around naturalist, and I sort of secretly wish I was an entomologist, but it's very hard. There is not a equivalent of the Sibley Guide to Birds for moths. It's very technical. They don't have common names, and so we need guys like Mark Mello. So Mark is Director Emeritus of the Lloyd Center for the Environment there in South Dartmouth. He was there for 35 years. He's got an MS degree in zoology, and his expertise is insects, especially butterflies and moths. You might know the book Butterflies Across Cape Cod that he co-authored with Tor Hansen. It's a great book, but also estuarine and freshwater ecology. And he's chair of the advisory committee to the state's Natural Heritage and Endangered Species Program. And so I am excited to hear about moths. So Mark, assuming you're out there, can you share your screen?
I'm here.
Tell us about the stuff.
Sharing, but nothing's coming up.
Seems to be the trend.
Like you are. Okay, open share tray, and why is my... Do you have the program up?
Nope.
No.
Joel, we could always have Joel share it if it, I think.
Okay, because it's up on my screen. It's not up on yours?
No. So share content, you find that share content button, and then...
Yeah, I clicked on share content.
And then click on whatever, the first window on the left is usually what I do of what comes up. And then go into your PowerPoint.
Nothing is coming up.
Joel, do you want to try, just in case?
I got screen share, and when I hit it, I get your Minimize it. Stop sharing your video when you started sharing.
Mark, I will go ahead and pull up your slides on my end,
Okay,
and you can just tell me when you'd like me to advance to the next slide.
okay. Can you make it full screen? There we go. Okay. Berry Beach moths and their role in conservation. Next slide. Why moths? One of the reasons it makes up 90% of the fourth largest order of insects, Lepidoptera. They're a major prey for neotropical migrant birds. Watch a nest of warblers or whatever, probably 9 out of 10 items that they're bringing to the nest are caterpillars. They're also major pollinators, but since they're pollinated at night, they don't get the attention that, say, butterflies and certainly bees. And in terms of how work with moths has also increased in the state, is because there's 37 species of moths on the Massachusetts endangered species list. And then there's aesthetics. Next. Most people think of moths as something that's various shades of fuscus, sort of gray brown. Next slide. But there are spectacular moths in the state. Polyphemus, next slide. And next slide. And then the Luna moth, put that up against any moth or butterfly from anywhere in the world for sort of grace and beauty. Next slide. And some of them are even fuzzy little microfauna. Next slide. Work of moths has really started taking off since 1992, which regulates the take of animals, plants for that matter. Two things, and I say regulates, not prohibits, because you can get permits to do a take. One I get in order to be able to track these things. We also protect habitat, and it's protected on private property. The only state in the country that actually protects habitat of state listed species on private property. So it's a very strong regulation. It's generated a lot of work. Next slide. Now, in terms of rarity, we talk about, rare species are on the list. But rarity is not a great term. This moth here, which is the water willow borer, a common name, as you can see, it's pretty widespread throughout southeastern Mass and what's left of the coastal plain. However, this is the global population of this species. It has its own its own endemic species. So that is protected as a category threatened, because even though it's relatively widespread, changes in climate could easily impact the whole population, considering how small it is. Next. This monster, the black witch, is very rare in Massachusetts. So it is not protected. It's subtropical, breeds worldwide in the subtropics and tropics. There's a strong flyer and does occasionally end up, I've seen 2 in my lifetime on tape. Next slide. So that's not protected. To be protected, you need to be breeding and native to the state. Rather than reading all these things, this is just sort of get a sense of the habitats of the moths on the state list. The lion's share are in barrens and heathlands, and that's a global rare habitat, so not surprised there's a lot of global species associated, globally rare species associated with it. You swing on over and you can see Barrier Dunes has 3 species that are affiliated with Barrier Dunes. Next. And in terms of the work that's gone on, the first project, first list was put together. There is no official Massachusetts list at the moment. first list that was put together was by Harrison Hitchcock, 387 species of moths. Farquhar did his PhD thesis at Harvard, and he documented 1,580 species of moths in the state. Kimball and Jones worked on Connecticut and Martha's Vineyard, and on Nantucket and Martha's Vineyard, had 1,300 plus species. Stephen Whitebread and I have trying to produce a standard state list with the help of a lot of contributors. We're already up to 2,800 species of moths in Massachusetts. So there's a lot of moths in the state. This work is largely due to the fact that there's a resurgence in basically field work, documenting distribution and ecology of species. Next. A wide barrier dunes, mainly because there's state listed species present and it's generally undersurveyed region-wide. There are 5 surveys that I've been involved in prior to the one that we did at Dead and Horseneck. But in these 5 was over 500 species of moths. So clearly there's a lot of moths that are occupying the area system. Next slide. So 2 sites. Picked Dead Neck, which is the mouth of Waukeet Bay. Next slide. Primarily early successional habitat, a little close stamp pine areas. Next slide. And Horseneck Reservation in Westport. And that's a much, much larger dune system. Next slide. There's basically Horseneck here, and it has fairly large wooded areas on what is still a sand barrier, sand dune system. This one is more akin to, say, Sandy Neck in Barnstable and Sandwich. Next slide. So the habitat surveyed at Dead Neck, it was primarily beach grass, herbaceous community, and maritime shrubland. Horseneck, the samples were mainly dwarf heathland shrub, acid shrub fen, altered shrub wetlands, and freshwater marsh. Much larger system. We didn't do the beach grass herbaceous system because Horseneck has about 10,000 people on the beach regularly every summer day, and we didn't want people sort of investigating what's going on with the traps, and I fear the traps may disappear for that number of people around. Next slide. So this is the trap that we brought out, and I hoof it out with a car battery. Next slide. Turn the light on. Next slide. Come back. Hopefully the light is still on. Look at the bucket. Pour out the moths. Next slide. And sort and identify. And then depending on the system that we were doing on the project, might actually have to count the number each. And I usually do just macro Lepidoptera because many of the micros you just can't identify by sight. Next slide. So this is comparison through dune complexes through 2019. The numbers of species varied pretty widely. Somewhat, but not necessarily relevant to the number of samples that were taken, more to the type of habitats present. But the listed species range from 2 to 4, not a large range in the number of state listed species, but in all the barrier dune systems surveyed, there's at least 2 species that are on the state species list. Right? And this is the number of stations in each habitat that we've looked at. You can see in most of the habitats, it's clearly that this graph is still rising. The number of species is still increasing with the number of stations that one looks at within each habitat. This is an odd artifact because it's randomized data and the highest number ended at first. But at any rate, there's a lot more to be found, basically, is what this graph is saying in any of the particular stations or habitats that we looked at. Next slide. I identified 18 species, which are listed here as being affiliated, particularly with dune habitat. Not necessarily restricted to dune habitats, but they apparently are breeding in dune habitats. I say affiliated. Well, you can think of this as a community. The whole thing is a community of barrier dunes. Big differences you can see in habitats, basically on food preference in these. But other than this odd thing here, the species is super abundant in most dune systems. I have no idea why it was so uncommon at Dead Neck. But if you look down here at these beach plum affiliates here, there's a decent number for three of the species, or actually four. Next slide. Okay, we'll look at a few of the species and sort of look at their range, both locally and range-wide. This is the pink streak, and its larvae feed at least on switchgrass. Next slide. This is these country maps are from Moth Photographers Group, and you can see at least in the Northeast, it's fairly restricted to the coast. Probably pothole prairie habitats out in the Midwest. Next slide. Now, locally through our Mass Moths program, you can see it's quite restricted to the coast. So here on the Camp Edwards Air Force Base, there are areas of sort of wet meadows that are dominated by grasses, lava post plant, and sure enough, we find it. That's well, just kind of mimicking the barrier dune system, even though it's not really barrier dune, it's outwash. Next slide. These species that are listed in the State Species Act. This one, pale green pinion moth, shrub feeder, particularly on hollies and other wetland shrubs. That is found at horseneck, not on dead neck. And the reason why it's not really a dunes affiliate, but if the dune system is big enough to have wetland habitats, you might find it present. These two species, Goldstein and Nelson, state entomologist, I have recently documented beach plum larval host plant. Next slide. And this is the Abigratus. Benjamin state has given it a common name, the coastal heathlands cutworm. Very, very narrow distribution. And it has a very similar species that's found throughout the West. Next slide. But as you can see, that with the various systems that I and others have looked at, again, it's very coastally related. And actually, there's a lot of beach plum, as well as a switchgrass in this area at Camp Edwards. More beach plum than you would expect as far as aerially. So it's also found there, but still a coastal species. And I would call it a coastal affiliate because it's not technically stuck right on the coast, right in barrier systems. Next slide. And this is a similar slide. It's a more widespread, again, right along the coast, right along the shores of Lake Michigan, and probably in pothole prairie habitats further out west. Next slide. And locally, again, a similar distribution to the cutworm. It is basically coastally oriented in Massachusetts. These are definitely dune affiliates. Next slide. And looking at the state list of species for all the habitats that have been looked at, and areas that have been looked at, these two species I mentioned earlier are found in all the habitats that are most abundant. Some of these were one species that is found. We really don't know whether it breeds there or not. When it's one or two species, as these things fly, we can't say for certain that we got a sustained breeding evaluation of those, but we certainly can for Pistis and Avenrose. Next slide. Now, other affiliates which are not listed, and I won't go through all the graphs of these, they're crazy, all the maps, crazy if I did. But we have other affiliates, Badia, which feeds on bayberry, and found on these dune systems. This species, Drastia graphica, feeds on the beach heather, Hudsonia tomentosa, which is abundant at the Horse Neck Reservation. We found numbers of these, but it's virtually absent at Neck. And surprisingly, we found one. We found any. This species very closely mirrors the two other beach plum feeding species that are listed, and this probably could be listed as well, even though it's sort of use protection, by virtue of the other two species on beach plum that are already protected. Next slide. Then forbs, sort of deciduous plants, low-growing plants such as composites. This is the sickle leaf silk grass, which this species, Shittia, breeds on. And this is abundant on dead neck. And I saw a lot of these fly during the daytime as well, and I saw a number of these flying around, as well as getting them in the light trap. This is a very, very xeric situation. The driest parts of dead neck, pairs of vegetation, carpeted with this plant, but will also grow on inland dunes as well. We do not know the food plant of this species, Jeremia, yet, but it's probably a composite. Most of them feed on various Asteraceae. And it is, again, affiliated with dunes. I find it in dune systems, but not restricted. This species, however, the seaside goldenrod borer, common name, is basically one that I would call an obligate. Next slide. This is the distribution that my photographers group folks records for. Next slide. We don't have it in Massachusetts, but we see it completely tied to coastal distribution. Again, it doesn't seem to feed on the other goldenrods, which are everywhere. It seems to be tied into seaside goldenrod, which may indicate that the dynamics of the habitat may be as or more important than the goldenrod itself. But this I would call an obligate species. This is one of probably a good candidate for this thing is rare. Next slide. Grasses, which not surprising in dune systems, you've got a lot of beach grass and other grasses growing there. These are all affiliated with those species. This one, maybe a Lintneri, and Extincta, at least in the Northeast, are almost dune obligate, feeding on the American beach grass. Next slide. Others, whoops, jumped on. Others on grasses and other forbs. This is Zoa. And this species, I had mentioned earlier, this was one that was super abundant at Horse Neck. Basically, this is a day flyer that you would see in September. Can we go back one more slide? These are Pamias. We don't know the food plant of these two yet. This whole genus, which is fairly large, the larvae feed on the roots of grasses and other low plants. And as a result, it's hard unless you go want to, tearing up, the habitat, you're not going to find larvae. So a lot of species in this group, the larvae have not found. It's sort of by accident when you're doing something else that you pull up something by the root, there'll be larvae in the root. Next slide. Actually, slides. And these are sort of visitors to dune systems. Widespread in distribution, not restricted to dunes, appear in dunes. Eumorpha feeds on a whole host of plants, Virginia creeper, grape. It's sort of like the birds that come into the coastal dune systems. If it finds its host plant there, this is actually at the northern edge of its range and has decreased over time in its occurrence in Massachusetts, but does now show up occasionally in dune systems. And if we've got a mild winter and it can find the host plant, and it's a female, of course, it may very well have at least a small colony for a year or two. This species, the wild cherry sphinx, when I was growing up in the 50s and 60s, I could find this in any suburban vacant lot in Bedford, that had cherry growing. I could find a larvae enough searching. This one again has really shrunk in its range, but we do find it in dune systems with either beach plum or cherry. So the barrier dunes are serving, sort of as a refuge, or at least a temporary refuge for a couple of these species, even though they're not obligate or really technically affiliated with dunes. They're just happy they're there. Next slide. So conclusions. Barrier dunes support a diverse moth fauna that is not yet fully known. And we're up with the work that I did in Rhode Island this year. We're up over 600 species of moths that we found in dune systems. Next slide. The habitats of deadnet supporting two state-listed species, the Heathland's cutworm and the Southeastern, the dune noctuid. Obviously, that was the name the state just gave to it because they have a common name. I'll say anything that's listed, apparently. And they feed on beach plum. So beach plum is an important plant species, the species that are listed in the Endangered Species Act. Next slide. And then, as I mentioned earlier, at least 15 additional moth species, dune affiliates, that are found at deadneck and are either grass, forb, or shrub feeders, larvae. Next slide. The threats to this habitat at deadneck, in particular, a storm overwash and non-native beach rose. I haven't seen any data on, competition between beach rose and beach plum, but in some areas that I've looked at, beach rose seems to have discovered areas that I would have thought would be colonized by beach plum. Next slide. So I think the takeaway from all of this is that, especially with sea level rise, climate change, dunes are going to get overwashed more frequently, even in, and need restoration efforts. And I think beach plum and bayberry, which also is host plant for the underwing that I've shown, and several other species, that these should be thought of as well as beach grass as plants to plant for restoration. And some areas, area dunes can be built on and are built on. Up in Massachusetts, I think most dunes are relatively well protected, but you can see like up at Newberry on that dune system, houses are literally falling into the sea that were built in dune systems. So not 100% protected from development if they're large enough. Next slide. Acknowledgements. I'd like to thank the Walkway Bay National Estuary Research Reserve and the Mass Department of Conservation and Recreation for permission to do work on their properties. Funding came from the William Watton Trust and the Beach Peak Trust. The National Heritage and Endangered Species provided me with the collecting permit to be able to do this and state-listed species. I also think in Harvard Museum of Comparative Zoology, which is where all these species will finally find their resting places in their collection for permanent future. And Stephen Whitebread for the slides you produced for those nice state slides. And I should mention my photographers who for the and of course the Lloyd Center, which is where I was working doing this program. And I believe that's it.
Great. Thank you, Mark.
You're welcome.
Good stuff. I like to see obscure insects and plants that don't get enough love getting some love, like goldenrods. Incidentally, goldenrods host more species of Lepidopteran larvae than any other herbs, any other flowering non-woody plants, basically, according to Doug Talamy work. Also, they don't cause hay fever. Okay, moving right along. We're running a little behind here, but getting right into Dr. Charlie Innis. Very happy to have our longtime partner at the New England Aquarium. You're probably familiar with our sea turtle rescue work, and those guys are doing the heavy lifting at the other end, really rehabilitating these turtles that we're getting off the beach. And so we've worked with Charlie for a long time. It would take me 10 minutes to read his illustrious biography here, arm's length list of accomplishments. He's the director of animal health at the New England Aquarium. He's also an adjunct professor at Tufts University, Cummings School of Veterinary Medicine, and various other international professional positions. Check out his bio in the abstract packet when you get a chance. Today he's going to be telling us about sea turtles released from rescue and rehab around the U.S., hopefully with a peek into some of our Cape Cod turtles. So looks like Charlie's ready to go. So take it, Charlie.
Thanks. Can you hear me?
Yes.
Can you see my slides?
Yes.
Is my slide advancing?
It is.
Okay, great. Thank you. So turtles have been around for a long time. They've colonized a lot of different environments around the world. Basically anywhere that it's warm enough for them to survive, they have evolved. And so we've got marine species, we've got freshwater species, we've got terrestrial species from the tropics all the way into temperate climates around the world. There's around 300 or so species of turtles. Their life history really relies on or allows for the low survival rate of eggs and low survival rate of hatchlings. But once turtles reach adulthood, at least prior to humans being on the planet, it was very likely that they would live for decades, human lifespans, if not longer, and be able to replace themselves before they die. So as you'll see through the presentation, turtles are really in trouble because of us killing adults. Their life history really does not tolerate loss of reproductively active adults from the population. 200 currently are listed under the IUCN Red List as various levels of conservation concern. Almost all of the sea turtle species are considered endangered globally. And in Massachusetts, including our sea turtles, the only species that are not state listed are snapping turtle, painted turtle, musk turtle, and the spotted turtle. And the spotted turtle is a pretty controversial one. So they were listed at one point and were delisted under some substantial pressure and protest a number of years ago. Bottom left is one of our beautiful native species, the wood turtle, taken out here in central Mass a couple of years back, and a Wellfleet eastern box turtle there, taken by Wellfleet volunteer Tim O'Brien. That turtle was medically treated a number of years ago, and Tim encountered it again some number of years later back at Wellfleet. The species we're mostly going to focus on today, the U.S. Common U.S. sea turtles, are upper left, the Kemp's Ridley sea turtle. It's the smallest and rarest sea turtle species globally. Their adults mostly reside in the Gulf of Mexico and nest on beaches of Texas and Mexico. But this is the most common species that we deal with as cold stunned in New England. The juveniles forage in this area in the summer, and it's about 90% of the turtles that strand on Cape Cod every year are Kemp's Ridley juveniles. This leatherback turtle in the upper right, this is a shot taken off the coast of Cape Cod by Cape veterinarian Tom Burns last summer. when he was diving looking for basking sharks. Amazing photo, amazing animal. Big adult leatherbacks forage off the coast of Canada and the Northeast US in the summertime, and we've got active research programs for that. Bottom left is the green turtle, the bottom right is the loggerhead turtle. Global threats to turtles in general include, in the past at least, hunting. A lot of hunting has been eliminated now due to legal protections. Eggs were hunted heavily in the past. Fishing interactions, as I'll show you in a minute. Vehicle trauma terrestrially by cars and in the ocean, vessel strike is common. We're seeing foreign bodies similar to what Lisa Sette described earlier. Disease is a problem for some species. And for sea turtles in particular, they tend to nest on beautiful tropical beaches, and these have been largely taken over by human development. And in some of the big oil spills around the world, sea turtles have been adversely affected as well. So I'm just going to show you some publication titles and local examples of some of these conservation issues. So watercraft, we see leatherbacks every summer. This is from Karen on the Cape. We see leatherbacks every year that have these really awful vessel strike injuries that are generally fatal. Sometimes we find them when they're still alive with horrible injuries like this, but they succumb pretty quickly. Entanglement is a big problem off the Cape with both whales and also leatherback turtles. We see a lot of entanglements every summer. Some of these turtles are already dead when we find them. Some have horrible injuries but are still alive. Others are disentangled successfully and released, and we do have evidence that disentangled leatherbacks, at least some of them, can survive in the long term. But some of these animals are alive with just awful injuries. The turtle in the bottom right was alive when we encountered it on the beach and was euthanized, obviously. But it's just amazing the level of injury that they can be alive with. Fishing industry, both through incidental capture and trawl nets and gill nets, etc., but also the longline industry. These photos are courtesy of Terry Norton, Georgia Sea Turtle Center, showing hooks in the stomach on the left and in the cardiac region on the right. Relatively recently, we've realized that sea turtles can be affected by decompression sickness, and this was probably a cryptic cause of mortality that was not recognized previously. Turtles can appear alive when they're pulled up from fishing gear and fishing nets. But if their blood vessels and major organs are full of gas, they will likely die after they're released. And this was first documented out of Europe, and we're now documenting this in a few cases in the US as well. As Lisa described, we see sea turtles ingesting plastic. This on the picture on the right was a case that Dr. Andy Stamper managed at Disney a number of years ago, and all that material came out of this one green turtle during its rehabilitation from intestinal obstruction. This is Dr. Brian Stacy recovering turtles during the Deepwater Horizon oil spill. We mounted a rehab response where these little juvenile turtles out of the Gulf were brought into rehab centers in New Orleans, treated and eventually released. And I think, as you can see, we're putting publications out about most of these things as well. So in addition to doing the rehab work, we're trying to document the physiologic status of these animals, the pathology that we see inside these animals. And we feel that in addition to just trying to release them back into the wild, another major importance of rehabilitation efforts is the learning that occurs and the publications that we get out, and training of students and training of veterinarians, public outreach, making people aware of the plight of sea turtles. This image of the Cape was taken by a physiologist from NASA out of the International Space Station a few years back. Mike Rapley is a turtle aficionado that lives in Texas. And I just wanted to use it to remind us of the shape of Cape Cod and that it actually is a pretty good turtle trap. Standora and Moriali and others have pretty well proven now that the Western North Atlantic is not just a freak place for turtles to show up randomly. It is a important foraging habitat in the summertime for leatherbacks, Kemp's Ridleys, loggerheads. And the Cape seems to be a trap where turtles that are foraging in the Gulf of Maine or in the bay itself don't seem to understand that they need to get north again to get out of the water. as it gets colder and head further south. So every year in the autumn, late autumn, we see turtles washing up on beaches of the Cape on the north side of the bay. And those are brought in through the Wellfleet groups to Mass Audubon, and then they're admitted either to National Marine Life Center in Buzzards Bay or the New England Aquarium. Beach walkers go out, volunteers go out, find these turtles on the beach, coordinated through Bob Prescott, Karen Dorderville. And the numbers are increasing over time. This is just a graph of turtles that were admitted to our hospital since 1999 that our colleague Adam Kennedy put together recently. So you can see it's really on an upward trajectory, and this was one of our biggest years this year as well. Lucas Griffin and others locally have published about this phenomenon. The numbers are increasing, and they're predicted to increase even further as the Gulf of Maine water temperatures continue to increase and provide good seasonal foraging habitats for sea turtles. There's a 2014 stranding season picture of Bob Prescott just trying to manage the hundreds and hundreds of turtles that were coming in on a daily basis to Mass Audubon Wellfleet. And this is a view of our sea turtle hospital in Quincy during 2014, when we were just trying to keep as many turtles alive as long as we could, and then sending them to other facilities around the US to complete their rehab. There's a lot of resources put into rehab, and a lot of resources put into sending them back out to the sea. The Coast Guard gets involved. You may have seen all of the cold stone work done in Texas recently, where big industrial ships were used to get those turtles back offshore into the Gulf of Mexico. So in wildlife rehab in general, it can be questioned whether putting all of these financial and human resources into rehabilitating the animals is really worth it. And some folks have called us out about this question. This was an opinion piece published a number of years ago that got us pretty fired up. A lot of these people are our friends and colleagues, but they're wondering, is it really worth doing rehab for all these turtles that are found stranded around the US every year? And especially we get questions about these Cape Cod juveniles. Isn't this a natural phenomenon? Shouldn't you just let them die naturally? Why are you putting all this effort into them? And we think there's all sorts of reasons why we should. It's our profession. There's a lot of other silly professions out there, and we think our profession is just as justified as anyone else's. We do believe that some percentage of these turtles are going to go on and survive and reproduce. And all of the other things I mentioned about the knowledge we gain for their medical management, basic physiology, basic biology, and outreach that we do to communicate about the plight of sea turtles is really important. So whether or not these animals are really living to survive long term is only part of the question, in our opinion. So based on that paper, it got me kind of fired up, and I put out a survey to all of the sea turtle centers that are permitted in the United States, and tried to put together a summary of the work that we've done and any evidence that the turtles are actually surviving after release. There's some limitations to how we can understand if they're surviving long term, and we'll talk about that later. Anyway, the paper was pretty good. It was chosen as the feature for this issue of Colonial Conservation Biology. We made the cover. These are our aquarium employees, Carrie McNally and Tina Wilkins, intubating and ventilating a cold stun Kemp's Ridley sea turtle. This is the outcome of that paper. We found that from the beginning of record keeping until 2016, there were over 11,000 turtles rehabbed and released again successfully to the wild. And only 300 of those were ever seen again. It seems like a low number, but as I'm going to talk about next, I think it's actually a pretty good number considering the needle in a haystack concept of trying to determine if you're seeing any of these turtles again. And 12 of those turtles, so roughly 1% of the 11,000, were seen successfully nesting again at some point in the future. So I think these are probably underestimates, and it provided some justification. And we did provide a lot of discussion in this paper about trying to gain a better understanding of how many turtles are actually reproducing in the future and how difficult that question actually is. So when we release turtles back into the ocean, the chances of ever seeing them ever again are quite low. They would need to encounter a human in some way to be reported. The males of sea turtles never come back to land. Females come back to land as adults every few years to nest. And someone would have to know that that female is one that went through a rehab program earlier in her life. And that question in itself is quite difficult when you think about how turtles can be using the entire planet if they wanted to. So we mark turtles when they get released. It's a requirement after rehab that turtles get marked with flipper tags. So in the upper left, we're putting on these metal flipper tags on the hind flippers. But studies have shown that flipper tags only last for a finite number of years before they fall off. The picture on the right, all the way on the right, is an X-ray of a turtle that has a microchip in its in its left front leg here. And we do microchip every turtle that gets released. This particular turtle was released, was stranded on Cape Cod in 2018, was rehabilitated at our colleagues in North Carolina, was released in the Gulf Stream off North Carolina, and was found stranded again, cold stone on Cape Cod this last fall in 2020. So we know that some of these turtles are surviving a number of years. We wish that they would figure out to go back into warmer places and not keep restranding, but it does happen from time to time. Anyway, the downside of microchips, even though they'll be in the turtle for their whole life, is that in order for someone to know that that turtle went through rehab previously, they need to either x-ray it and see that there's a chip there, or they need to interrogate the chip with a scanner on a nesting beach decades later. And we know that for Kemp's Ridley's nesting beach coverage in Mexico, which is their major nesting area, is quite low, at least as far as the ability to scan every single nesting female for the presence of a microchip. We can satellite tag turtles, but the batteries on satellite tags are short-lived. The map in the center is a leatherback that was disentangled off Plymouth last October that we've tagged, and the date of the final transmission down way in the south is 2 days ago. But this turtle's battery is going to die eventually. And since we are releasing a lot of juvenile turtles, we really need to know that they're reaching adulthood in order to prove anything about our conservation impact. And so 1 or 2 years of information for a juvenile Kemp's Ridley that's released is not really adequate. We need to know that they're still alive 5 years later, 10 years later. And the best way to do that, we think at this point, is to start to think about using acoustic tagging. I know Megan's going to talk about white shark acoustic tags later, so I won't go into too much detail. But there's a widely distributed array of acoustic receivers along the coasts of the U.S. And if an animal is carrying an acoustic tag, receivers will detect that animal and report to the researcher that owns that receiver, and then we know where they are. Turtles have been tagged with external acoustic tags for a long time. But they tend to fall off. Turtles go through pretty aggressive mating behaviors, and so any sort of external tag attachment is also limited. And we're finding that tags are falling off after a year or two, even though their battery might allow them to survive for 10 years. We know from our colleagues that study other species that internally implanted tags is a reasonable concept. And we've not yet been permitted by the feds to put in internal tags in sea turtles. We're arguing about that because they are permitting lots of tags to be put into many other protected species. Bottom publication was from Jeff Kneebone, our aquarium colleague, from 2012, but some of the sharks described in this paper are still transmitting 10 years after he implanted the tags. So we really are seeking permission to put in internal tags in sea turtles. It's much cheaper than satellite telemetry. The battery life is much longer. And if we can, 5 years from now, if we've tagged hundreds of turtles internally, we can really start to get a sense of whether they're surviving long-term over the next few decades. We have a huge number of partners that contributed to the survey and also that contribute to the rehab work here that happens every year. So thanks for listening, and you can contact me offline if you have questions. I know this is a huge realm of conversation and it raises tons of questions, so I'd be happy to talk about it anytime.
Wow, applause. How did that even happen?
That was
Thank you,
symbolic
Charlie.
applause.
Thanks very much, Charlie. That's good stuff. Okay, so now is time for our Q&A. It doesn't look like there are very many questions, which is good because we're way behind. Oh, here they come. Everybody waited. So we did have one question if the speakers, the first 4 speakers can have your video and microphones on. There was a question for Mark Mello about the status of the Moths of Massachusetts list with Stephen Whitebread and where people could access that.
Okay, can you hear me?
Yes.
Okay, great. Just recently, the county list of the species that we're working on has been put online. And you could access it by typing in HTTPS / all lowercase MASSMOTHS.org.
Moths.org. Look at that. It worked.
Okay. Do you want to
All
go to find a printer?
So it looks like Charlie has been answering some of the questions in the chat, which is great. But we can also read those out loud. Have you considered tattooing sea turtles with IDs as a way to ID them from boats? And Charlie, you can elaborate on your answer if you like.
Yeah, I guess.
No, it's just, it hasn't been really thoroughly worked on. I would wonder about the long-term visibility of tattoos as turtles grow out and slough skin and etc. But it's something that could be considered. Whether or not you would have unique ID numbers that were tattooed, I think is a question, or is it just IDing a cohort of turtles? So you know that it went through rehab somewhere in the world because it has a tattoo. But no, it really hasn't been discussed. Why are acoustic tags not allowed yet? I don't know. Call up U.S. Fish and Wildlife and ask them why they're allowing it for sturgeon and seals and other things, but no sea turtles yet. I think there's some perception that it's going to cause harm, and it's just never been done yet, but it's done in all sorts of other taxa.
Okay, thank you. Anything else? Boy, when this is live, people are asking all kinds of questions. All right. Not seeing anything else. We may need to just take take this extra time because we're way behind. We could always please write the access to. All right, let me do that quickly. I have that. I'll put the link to the massmoths.org in the chat here. Everybody can see that. Screaming babies here. All right. So we're gonna go right into Danielle Perry. Hopefully Danielle is ready. Try to pick up some time that we've lost here. So we're very lucky to have Danielle Perry on board here at Mass Audubon, looking at climate change, ecology, climate change, resiliency across sanctuaries, which is something we didn't have before, allowing us to take a more systematic and collaborative approach across the whole organization, looking at climate change and all of its impacts, looking for grants. Oh, look, we're getting some questions coming in, but it's too late. So she's our climate change adaptation ecologist. Dr. Danielle Perry. She works to enhance the resiliency of Massachusetts lands to climate change. So she's evaluating climate vulnerability of our sanctuaries, and that's what she'll be talking about today. Some very important work that matters a lot to those of us at the sanctuary level as we look at what will be happening to us with sea level rise and increased storms and everything else. So Danielle looks like she's all ready to go. So without further ado.
Thanks, Mark. Can you hear me?
Yes, you're good to go.
Is it still morning? So good morning, everyone. Thanks for joining. So as Mark said, I'm hearing some feedback.
Danielle, if you could just unmute yourself, we had to do a nuclear option and mute everybody.
Right,
Danielle, there you go. Thank you.
Let's reshare. Okay, perfect. So as Mark said, I'm working on completing climate vulnerability assessments of the Mass Audubon sanctuaries, and then to get a better idea of how they're expected to fare when it comes to climate change effects such as sea level rise, which is particularly important for our coastal sanctuaries like Barnstable Great Marsh, Long Pasture, and Wellfleet Bay. So I started this process with looking at 5 pilot sites, so 5 different sanctuaries of Mass Audubon, and I focused first on Joppa Flats, Rough Meadows, and Eastern Point, which is located in our North Shore area, and then also with Barnstable, Great Marsh, and Wellfleet Bay, and I'll be sharing the results of those Cape Cod sanctuaries today. And then just to get a better idea of location, because I think we're very familiar with our sanctuaries on the Cape, but just to get an idea, these are the North Shore sanctuaries that I focused on for the 5 pilot site vulnerability assessments. And so as I mentioned, I'll just be focusing on Barnstable, Great Marsh, and Wellfleet Bay to get a closer look at our Cape Cod sanctuaries. So these assessments were broken into 3 different parts. So the first part was being able to assess the resources at each of our sanctuaries to get a better idea of how we value them to accomplish Mass Audubon's mission as well as the sanctuary missions. And then it will help us prioritize those resources. And so we know where to act first and which of our sanctuaries are the most vulnerable to climate change and sea level rise effects. And then the second part is looking at a sea level rise assessment. So this is using sea level affecting marshes model, or SLAM modeling, in order to determine how our coastal areas are going to be changing with sea level rise, especially our natural habitats. And then also using data provided by NOAA to, again, get a better idea of how sea level rise and flooding is expected to impact our sanctuaries. And then the last part is looking at field surveys, salt marsh field surveys, and to get a better idea of the current conditions of our salt marshes, because these are really our first line of defense against sea level rise. And they're also an important mitigation habitat to help slow down the effects of sea level rise, because they do store a lot of carbon and actually store more carbon per unit area compared to forests. So salt marshes are an important adaptation resource and mitigation resource. And there really are our first line of defense, helping to mitigate flooding, helping to buffer our coastline and protect our roads and our buildings. So wanting to get a better idea of how our current conditions of our salt marshes are, which will then give us more insight into how vulnerable or resilient our properties may be. So like I mentioned, the first part is assessing our resources and giving them values. And this was completed using a resource prioritization survey. And so we built off of a survey that the trustees of reservations used for their own climate vulnerability assessments. And so for each resource, we ranked against these different categories. So looking at the ecological importance, the cultural importance, visitor experience, programming, financial, and operations, and then scaled that from 0 to 5. So 0 being least not as valuable for that particular criteria, 5 being very valuable for that particular criteria. And just to get a closer look at what our survey looked like, so our main categories here, and then we had these subcategories that we ranked each of the resources against to get a better idea of how important that resource was for these different criteria. And to provide examples of those resources, so we would rank things like our trail systems, our observation decks, our visitor centers, and of course, all of our natural habitats within the sanctuary. And we would use the expertise from our sanctuary staff as well as Mass Audubon scientists in order to rank these resources. And then each resource would then get a resource value score. And the second part of the assessments was the sea level rise assessment analysis. So this was more about GIS modeling. So this is just a photo of our Rough Meadows property, and each of the different colors represent a different natural habitat. And then I would overlay these images with infrastructure. So then we would also do infrastructure mapping. And I would use GIS to then determine how vulnerable our habitats as well as our infrastructure were to the intermediate high sea level rise scenario for the years of 2030 and 2050. And what would result is an overall impact of sea level rise score. And this score was encompassed by two separate scores, a habitat score and a feature score. So the habitat score would look at the percent change in habitat from current conditions until 2030 or 2050. So how our habitats were changing, and then multiplying that value by the resource score that was determined during those resource prioritization meetings. And then our feature score would then look at if that particular resource hard infrastructure was expected to be flooded in either 2030 or 2050. And this was all done using sea level affecting marshes model and NOAA sea level rise data. And so then I would just sum the habitat score with the feature scores to get an overall impact of sea level rise score for that particular sanctuary. And we would use this score to then prioritize our sanctuaries. So the scores that appeared to be more negative represented our sanctuaries that were more vulnerable to sea level rise. And just to provide an example, so this is an example for one of our northern shore sites at Joppa Flats. So some of this habitats at Joppa Flats is salt marsh and tidal flat. So our salt marshes were expected to lose about 6.4% of our salt marsh in 2030. So then we would just multiply negative 0.064 times that resource value of the salt marsh 27 to get a habitat score of negative 1.7. And then the same we would do for our tidal flat. And for our infrastructure, we would then, the main infrastructure being the road that kind of borders that sanctuary, which was expected to be irregularly flooded, so it only lose half of its value. And if it was regularly flooded, it would lose the full 29 value. So we have a negative 14.5 because it's only expected to be irregularly flooded by 2030. And then for the overall impact of sea level rise score, I would just sum up each of the habitat scores here with our feature score to get this overall impact of sea level rise score. So then again, we would use that score to then determine which habitats are more vulnerable than others to sea level rise effects. And so the last part of this assessment was then field surveys to get a better idea of current salt marsh conditions, site resiliency, as well as migration potential. So our resilience score, we would rank it from very low to high, and we would look at the disturbances on site to then get a better idea of overall site resilience. So some of the disturbances that we would look on site is nutrient inputs, ponding and plant die-off areas within our salt marshes, ditching within our salt marshes, as well as if there's invasive species present, which all are impacting resilience and account for disturbances. So the higher the disturbance, the lower the resilience score. And then we would complete a salt marsh conditions survey to then get a better idea of the overall habitat quality of our salt marshes. And so the higher the habitat quality, the higher the resilience of that sanctuary, and the lower the habitat quality, the more vulnerable the sanctuary would be to further sea level rise effects. And then our last part was the migration potential. So then looking at to see if our salt marshes have room to migrate backwards, if there's space for them to move more landward, and if there's active barriers. Because if there is space for them to move backwards, then that adds to the resilience of the sanctuary, because that will be the natural response for salt marshes to adapt to sea level rise. So marshes that would have more space to move landward would have a higher migration potential, and then marshes with more barriers preventing migration would have a lower migration potential. So again, these were our pilot sites, but I'm just going to be focusing on showing the results for our Barnstable, Great Marsh, and Wellfleet Bay sanctuaries, and starting off with Barnstable, Great Marsh in Barnstable, Cape Cod. So when looking at our resilience, it showed to have more of a moderate resilience, and some of the disturbances that we saw on site was the frequency of crab burrow densities, noticing some ponding and die-off depressions within the marsh, as well as ditching on site. And then, so for our salt marsh condition score, some of the vulnerabilities that we're noticing within our salt marshes is we're already seeing some displacement of high marsh plants by low marsh plants, which is an effect of sea level rise because high marsh plants are not as tolerant of flooding as low marsh plants. And because they're not as tolerant, we're actually losing our high marsh habitat and it's being replaced by low marsh habitat, which is a particular concern because certain wildlife species like the salt marsh sparrow is very reliant on high marsh habitat. So as that is losing, it's making our salt marsh sparrows even more vulnerable. And also seeing some die-off and bare depressions within our that particular salt marsh as well. But some positives for this marsh environment is that it does have extensive high marsh habitat. So even though we are seeing some displacement, there's still a significant amount of high marsh habitat on site, as well as being salt marsh shrubs within the property too. Then, as I mentioned before, so we prioritize our sanctuaries by this impact of sea level rise score. And more negative means more vulnerable to sea level rise. And so, of the 5 pilot sites that we looked at, Barnstable Great Marsh showed to be the most resilient because it also has a high migration potential. So it is surrounded by more natural systems. So there is space for the marsh to move backwards to add to that buffer. So the marsh to adapt to sea level rise effects. And another positive with Barnstable Great Marsh is that a lot of the infrastructure is more upland than right on the coast, which you can kind of see with our feature score is that there's not too much of a feature impact. I think this is just noting flooding of a particular bench within the habitat. So again, not too concerning for infrastructure. And then we're actually seeing a positive gain in habitat because as I mentioned before, this area has a high migration potential. So we're actually gaining habitat because it's able to migrate more landward because it has that extra buffer of natural systems. So our actions for Barnstable Great Marsh were just to build that resilience, to make sure it remains resilient to sea level rise. So one action that we could do is doing some ditch remediation on site, because ditches have shown, historic ditches, which has been dug in the past for like agricultural purposes, for mosquito abatement purposes, has actually shown to cause marsh subsidence. So wanting to do some ditch remediation in order to prevent marsh subsidence, because we want to be able to gain salt marsh capital and gain height within our salt marshes and not lose height. So they become more resilient to sea level rise effects. Then in Wellfleet Bay, so this site showed to have more of low to moderate resilience. And some of the disturbances that we're seeing within this site is crab burrow density, high frequency of crab burrow density. We're seeing some evidence of nutrient inputs, which can be kind of speed up the degradation of our salt marshes, as well as some extensive ponding and die-off depressions and soil disturbances on site. So there was a higher amount of disturbances compared to Barnstable Great Marsh. And so for our salt marsh conditions, some of the vulnerabilities that we were seeing was that there are bare sediment areas within our salt marsh and those die-off bare depressions as well within the salt marsh system. But some positives is that we're seeing distinctive high marsh area and low marsh area. So for our impact of sea level rise score, so out of the 5 pilot sites that we looked at, this site seemed to be the most vulnerable to sea level rise of the 5. So this is our first priority action because we're seeing a lot of vulnerable infrastructure as well as vulnerable natural communities. So for 2030, we're showing to have a higher impact of a sea level rise score to the -6 that we saw for Barnstable Great Marsh. And so our habitat score, we're not seeing too much changes as of yet, but for our future score, we are seeing some impacts of some of our buildings, our bird blind buildings, our trails, and which is making it vulnerable to sea level rise effects with their current locations. But when we jump to 2050, that's when we start seeing some negative impacts and loss of our natural habitats, especially because one of our freshwater habitats within Wellfleet is expected to become more of a salty habitat. So losing freshwater habitat and then having more of the infrastructure being impacted by sea level rise. So some things are becoming from irregularly flooded in 2030 to be regularly flooded in 2050, which making them more vulnerable and susceptible to sea level rise effects. And with our migration potential, we are seeing it's more moderate versus the high that we saw in Barnstable Great Marsh, because even though there are areas that are surrounded by natural communities, we also are seeing some steep slopes surrounding our salt marshes. And with the steep slopes, it is preventing marsh migration because the marshes can't really jump that slope in order to migrate backwards. So it's kind of hindering any landward movement of the marshes. And then I have some king tide photos of the Wellfleet Sanctuary just wanting to share to kind of be able to better picture some of the vulnerabilities and potentially what we might be looking at with sea level rise over time. As you can see, like our trail system being flooded here. And this is king tides from this, I think, November 2020. So some of the flooding impacts that we're already noticing during king tide. So these just want to make it clear, this is not normal tides right now. These are just special king tides that we're seeing, but it kind of gives us an insight of what the future sea level rise effects could be. Again, seeing that flooding of the trails within Wellfleet. And then these are our sea level rise markers. So again, king tides kind of already encompassing what we expect to see average high tide by 2050, even going beyond that point. So for some climate adaptation actions of wanting to potentially relocate some of our vulnerable trails, also wanting to look into the use of a living shoreline in order to mitigate some of that flooding aspect, to mitigate some of the salt marsh erosion and the wave energy that is coming into the sanctuary to help prevent some of this loss in habitat that we are expected to see. And then just to get a better idea of how our 2 Cape Cod sites compare to the other 5 sites that we looked at. So this dark blue being Wellfleet and Barnstable Great Marsh being in a striped color, and these are 3 North Shore sites. So as I mentioned before, Wellfleet for both 2030 and 2050 seem to be the most vulnerable to sea level rise compared to the other 5 sites, with Barnstable Great Marsh having the least negative impact of sea level rise score and being a little bit more resilient compared to these other sites. So again, the more negative, the more vulnerable. And so that is the end of my presentation. And then I'll hand it back to you, Mark.
Great. Thank you, Danielle. All right. So, starting to get a little action in the chat. Next up, who doesn't love a shark talk? And so we're very happy that Megan Winton is with us from the Atlantic White Shark Conservancy. Megan, she's the research scientist there, which is a Chatham-based nonprofit, as I'm sure you all know. She's a grad student at UMass Dartmouth. She's pursuing her PhD in marine science and technology. And since 2015, she's been working with Mass DMF to improve our understanding of Cape Cod's burgeoning and emerging white shark population.
Thanks, Mark. Can you see my my PowerPoint?
So far, so good. It's not in. Oh, there you go.
Okay,
Yeah.
I just want to make sure. I unfortunately have kind of a hard stop at noon, so I want to make sure I put it in presenter view so I can keep track of time and prioritize if need be.
Okay.
Is that it's still full screen for you, not presenter view?
Looks great. Go for it.
Okay, awesome. So I will Sorry that I got to get out of here at noon, but I will, if any of you guys have questions, if you just Google the Atlantic White Shark Conservancy, you'll be directed to our info box, or you can find my email that way too. So please follow up if you have any questions. I love answering shark questions, so I'm bummed I'm going to have to get out before the Q&A session. But again, hi everybody, I'm Megan. I'm a research scientist at the Atlantic White Shark Conservancy, which, as Mark introduced, is a nonprofit. We're based out of Chatham, and we were founded in 2012 to support white shark research off the Cape, provide the results of that research to town and beach managers so that it can be used to inform public safety practices related to the emerging population of white sharks, and to educate the public about their presence here off of our coastline. And that little head in the white hat at the bottom of the photo there, that is also Megan. That's me when I first started out as a brand new PhD student. I was out on one of my first ever research trips. About 6 months earlier, I had started my PhD at the School for Marine Science and Technology at UMass Dartmouth, where my dissertation research was, and still is, I'm getting close to the end here, but not quite wrapped up yet, is focused on estimating the size of the white shark population off of our coastline. To do that work, I was and still am working really closely with Dr. Greg Scommel, who's at the top of the photo there, at the Massachusetts Division of Marine Fisheries, who started the Massachusetts Shark Research Program and has been studying white sharks in our waters longer than anyone else. And he's really brought our understanding of these animals a long way. Next to him on the pulpit there is Cynthia Wigren, who founded the Conservancy. She quit her day job to start the organization to help fund white shark research when she learned about how tough it was to find funding for white shark research, which is a whole story in itself. And so the two of them are out on the pulpit. They're both pointing out a big shark, which you probably see. That's a 15-foot long female white shark nicknamed Freckles. You'll hear more about her later. And for me, I had wanted to be a shark scientist for a really long time. So this was a huge moment for me. I look pretty calm in that photo, but I was pretty much freaking out. I had a lot of emotions going on. On the one hand, I was so excited to see this animal, this species I had spent so much time reading and thinking about. It was honestly kind of like seeing a mythical creature to me at that point, like seeing a unicorn. But it also, to be honest, was kind of a sobering moment. We were right off of, we were really close to a beach in Chatham, and this was a 15-foot long, 2,000-pound plus predator. And so it made me realize how important the research we're doing here is for the white shark population, but also for our community, for Cape Cod ocean users. So that was a really big, memorable day in my life, I gotta say. And the reason that this is also important is because up until the last few years, we really knew next to nothing about the species in the Northwest Atlantic, other than the fact that they were a really elusive, long-lived predator that had been in serious trouble. Based on fisheries catch data, we think their population declined by as much as 80% from the 1960s into the 1990s as a result of fishing pressure. But scientists had never been able to conduct any type of standardized survey to estimate the actual size of the population, which is really critical information when you're trying to determine the status of a population and figuring out whether it's recovering, declining, how is it doing. So that's really important information from a fisheries management perspective. And because Cape Cod is now an aggregation site where white sharks come in the summer and the fall to feed on the recovering seal population, we had an opportunity to estimate the size of the population for the first time. And the answer to the question, how many white sharks are out there, which is the question we get by far the most often, it also has really big implications for the beach-going public. My guess is that if I tell you that 5 white sharks are coming to Cape Cod to hunt for seals every year, you are going to use the water differently than if I tell you that hundreds of white sharks are coming to the waters off of Cape Cod every year. And so even though this was one of my first research trips, it was the first time I ever saw a white shark, I had spent a lot of time in the previous months reading up on how we would be coming up with this population estimate using what are known as mark recapture methods. So we typically can't go out and count all the individuals of a species in a given area, but there are different methods out there that allow us to estimate the size of the population based on what we can observe. And so mark recapture methods they entail going out, you sample a certain segment of the population, you mark the individuals you see, you then quote unquote release them back into the population, and then you go sampling again at a later date and see how many of the previously marked individuals you encounter on your next sampling day. And by calculating what proportion of the second captures are marked, which we call recaptures, we can come up with an estimate of population size. And it's really kind of an intuitive thing. It seems kind of weird, but the idea behind it really is that if there are a lot of sharks out there, odds are the next time we go out, we won't see that many previously marked individuals because there are so many different sharks out there. On the other hand, if there aren't many sharks around, the proportion of recaptures is likely to be higher, and we'll just be seeing the same individuals over and over again. So in order to apply these methods, we have to be able to uniquely identify each individual. Sometimes this is done by applying a mark, such as a tag. But as I knew from all the papers I had been reading leading up to this, in the case of white sharks, we can identify them based on their physical characteristics. So on each trip, the idea was we would go out, get videos of sharks, and mark them based on their unique physical features. And just holler if this video doesn't start playing. Hopefully it works. Oh no, it's making noises. Well, you'll just hear some water noises. But so when we got off the water that day, I was so excited to review the footage and see if I could mark my first shark, if I could identify the shark. So this is the footage of that encounter. You'll see Greg turned on the GoPro and he just kind of sticks the pole in the water as we approach the shark to get underwater footage of the animal. At this point, I'd watched a lot of shark documentaries. I thought I'd be like really good at this, but when I saw that first clip you guys thought, I just was like, oh my gosh, it's a shark. It's actually a white shark. It might sound funny, but you know, actually seeing them underwater, seeing that underwater footage made me realize that was actually, even though I knew that was out there, but with each pass of footage, I started to notice more and more details. You can see the unique colorations along the body, patches, you can see the fin shape. And so really the more of these videos you watch, the more these individuals start to look really different. The easier it is to tell them apart just by eye. And I apologize for what I'm presenting today. You can't unhear this information, you can't unsee this, and it will probably ruin shark documentaries for you forever because you'll realize that what they're calling the same individual is. So the feature that you just saw there, and which is the fin on the back of the animal, that is the feature that is most commonly used to ID white sharks in all regions of the world where white sharks aggregate. So the back side of the dorsal fin, which we refer to as the trailing edge, you'll see has a series of notches that are unique to each shark. You can really think of it as kind of like a fingerprint. You can also see that some of the sharks in this slide have white patches on their dorsal fin, which are called rosies, which we can also use to help identify these individuals. And if any of you guys have a keen eye for sharks already, you might have noticed that there's a familiar dorsal fin in this slide. So that one that's outlined in pink, that is Freckles' dorsal fin. And again, this is a really ideal feature to use, not just because it's distinct, but because the notches are the same from both sides. But we aren't always able to get a good shot of it because of the method we use to collect our footage. It's really hard to key in on just the dorsal fin. So we work with a spotter pilot who flies around and finds sharks for us off the coast of the Cape. It's really a great place to do this. The beautiful white sandy bottom off of our coastline provides really great contrast with the body of the animal, as you can see in that lower panel picture. And so our spotter pilot finds sharks. He basically just guides us to them, and then we sneak up on them and get underwater video footage. I have another video, but it has music and it's going to be loud. And since I want to get to some of the other stuff, so I'm not going to play this video right now, but we have an amazing captain who does a really great job sort of dancing with these animals to allow us to get video footage, but it's still a really hard thing to do. These sharks have moves. So because we don't always get a good shot of the dorsal fin in our footage, we have to use a lot of other characteristics. And luckily for us, white sharks have very unique pigment patterns along their bodies that allow us to determine individuals, even if we don't get a good shot of the dorsal fin. So people don't think of white sharks as really like colorful, beautiful animals usually, but they have really intricate, unique patterns on their tails and along their bodies. When you look at this, so this is a shot of a bunch of different pigment patterns on tails of different white sharks we've identified off the coast. And this is how a lot of them get their nicknames. They all get an official numeric ID, but it's really hard to keep track of them just thinking of a bunch of numbers floating around in your head. So they all do get a nickname as well. And a lot of them get their nickname based on what these spots on their tails look like. The more you look at these, the more it kind of becomes like looking at clouds and imagining shapes in clouds. And again, there's Freckles' tail. I don't know if any of you picked that out right off the bat. If you did, please contact me. We're always looking for members of our Shark ID squad. It's a ton of work. We also use pigment patterns on the gills. They're also very unique to individuals, but how far the white extends along the gills, if there's any unique freckles or patterns. And that is how Freckles, who again is outlined in pink here, that's Freckles, the right side of her face. That's how she got her nickname. She had very freckly gills in comparison to a lot of the other sharks we encountered. So it might seem silly, it might seem not very scientific to give these animals nicknames, but by assigning them a nickname that relates to a physical feature, it really helps jog our memory when we're going through all this footage, identifying individuals. So we go through and we classify each side of the shark, these different body regions according to some common patterns, and we use that to catalog every individual we've encountered. It sounds like a really fun matching game, but it is sometimes, it can be incredibly time consuming and tedious. We essentially have to go through this footage frame by frame. So for the population study, for the mark recapture study, that's part of my PhD work, we collected over 3,300 videos from 2014 to 2018. So far, we've documented 509 potential individuals. And why I call them potential individuals is because they're unique. They don't match to other sharks in the catalog, but for all of those individuals, we don't have both sides of the body. I wish that they were symmetrically colored. It would make our job a lot easier, but each side of the shark and each region is different. So we need to get both sides of the animal to confirm it's a unique individual. So we have 509 potential individuals in the catalog right now. We'll be using that as the basis for coming up with our population estimate over the next couple of months. I wish I had one for you today, but we're getting there. But one thing I just want to mention is that, we were out there to catalog white sharks, to generate a data set to provide a basis for estimating the population size of white sharks. But we've learned so much more about these animals than I think any of us initially anticipated from this footage. It's really given us a glimpse into their lives. So they are the only natural predators of seals in our area, and we're learning a ton about predator-prey relationships from this video data set. We've been able to collect direct observations of predation events on seals, as you see in the lower left-hand corner there. That shark actually, and we've seen this several times, they'll get a seal, they'll hook the carcass with their teeth, and they'll head offshore. And we think that's to avoid another shark coming in and trying to steal their meal. It's a really tough thing to catch a seal. They're very agile, fierce predators in their own right. So you may have noticed in the video of Freckles that she had a lot of scratch marks on her face. You can see scratch marks in those top two photos on this slide here. And those are actually scratch marks from seals fighting back. They have big claws, they have teeth. You can see in that top right photo, that seal really was targeting the eye region of the shark. So they can inflict a fair amount of damage on these animals. So we also see other indirect evidence aside from seal scratches. In that bottom right photo there, that's intestine hanging out of the gill. I know that's kind of gory, but from our perspective, it's really valuable information. We know that shark ate a seal not that long ago. So by looking at patterns and healing rates of seal screeches, trying to go too fast because I know I have limited time. But this is giving us insight into their predatory habits, and it's allowing us to better understand how they might be impacting the local seal population through predation pressure. We've also learned a lot about how they interact with each other. They do have social lives, not really the way we think of them, but it's something not a lot of people realize. We often see sharks with bite wounds. For smaller, immature sharks, we think this is related to dominance effects. They get in the way of a bigger shark, and a bigger shark isn't having it. For larger sharks, such as Freckles, bite wounds can be evidence of mating activity. Male sharks have to hold on to females for mating. So when we see bite marks on a shark the size of Freckles, who at 15 feet and roughly 35 years old is only right now at the size at which she's maturing and is only now going to be starting to reproduce. So those types of bites can be indicative of mating activity. And so, based on her size, we think Freckles is approximately 35 years old. I'm 36. I've been around for a while, and it really makes it hard not to kind of start to wonder about the individual lives of these animals and the types of things they've had to navigate in order to make it to the size and the age where they can even reproduce. The ocean is a really tough environment. We tend to think of white sharks as these big, fierce, invincible predators, but the reality of it is they're just big fish, and it's not easy out there. They spend a lot of time in nearshore coastal waters. They're vulnerable to boat strikes, just like whales and sea turtles. So that photo in the top left there, that's a propeller wound going down the back of a white shark. They're a prohibited species. They have been since 1997, which means you can't go out and harvest white sharks, but it doesn't mean that they're immune to fishing gear. We've documented a lot of evidence of interactions with different gear types. The top right corner, there are deep cut marks and the tail is severed. We think that shark was entangled in a line. The bottom left, the shark has a fishing lure attached to its fin, probably from when it went into to steal a fish from the end of a fisherman's line. And the shark at the bottom right has some fishing line coming out of its gills, which is kind of abrading and aggravating that region. But we've been following some of these individuals for years. Some have come to Cape Cod every year for the past 7 years. It's incredible from a scientific perspective to follow a fish for that long. And we're really getting a better understanding of this mysterious species. But it's also, I'm a scientist, but I'm a person. And it's also, it makes it really hard not to start to think of these sharks on an individual level and kind of emphasize with their reality. And you really start to think of them as something other than the mindless eating machines that they're often presented as in films and documentaries. I wish I had more time to tell you more. There's so much incredible information. I'd love to tell you more about some of these individual sharks. So please reach out. Like I said, if you have questions, you're probably wondering if we've ever seen Freckles again. We haven't. It's something, I wonder about her a lot. I wonder if she matured, she just moved to a different area. She's off wandering the broader Northwest Atlantic. Did something happen to her? Has she been back to the Cape and we just haven't encountered her again? So I wonder about her a lot and what's happened to her. And please, like I said, please reach out if you have any questions. Thanks so much for the invitation to be here today.
Very good. Thank you, Megan. Freckles. I love it. Oh, cute widow Freckles. I love that there's something out there named Freckles that's dismembering 800-pound gray seals. So it sounds like you can't stick around for questions, but people know where you live and work, and they can probably find you there. So
it's easy to find in Google, but please, I love to answer shark questions. I'm so bummed that I have to run. So please don't hesitate and don't think you're bothering me or anything. I would love to share more about this work. And again, thanks so much for the opportunity to present today.
Great. Thank you again. Okay. The anchor leg of our race here today is Jason Zimmer, in our charismatic megafauna section of the day. So Jason will be talking to us about bald eagles, something that is on a lot of people's minds here on the Cape as the population has been burgeoning, and we're starting to become aware that they may just be nesting here cryptically. So Jason's a Southeast District supervisor with Mass Wildlife, has been for 16 years. I've worked with him in the past on plovers on the Outer Cape. He's a Southeastern Mass native. But Jason has done the kind of wildlife research that every little boy in a wildlife management program at college wants to do. He's worked with elk and mule deer and grizzly bears and mountain lions out west, which is what we all dream of. But now he's back home here in Southeastern Mass, where people are afraid of 10-pound weasels called fishers. So it's a little different here, but very happy to have Jason here to tell us everything we want to know about the bald eagle recovery in Massachusetts, with a special focus on Cape Cod. So Jason, if you want to share your screen. If you're out there. He is, look at that. Looking good.
All right. Can you see it?
And I can see it and can hear you, so you're good to go.
Great. So thank you for having me. I appreciate being able to be a part of this conference. I'm not necessarily presenting like a research project. It's more of a story of the bald eagle and history of the bald eagle in North America. But I'll also provide some information on what they've been doing in Massachusetts and on Cape Cod in particular. I think Mark mentioned earlier when he was talking about moths that some species don't get the respect they deserve. Bald eagles are not one of those species. They get tons of attention, tons of respect. They're kind of one of those universally revered species. So it's one of the fun ones that I get to work on here in Massachusetts. Before I get started talking about eagles, I'll just tell you a little bit about our agency. I work for Massachusetts Division of Fisheries and Wildlife, or Mass Wildlife as we're commonly referred to. And we are the state agency that's charged with the stewardship of all wild amphibians, reptiles, birds, terrestrial mammals, our freshwater and anadromous fishes, and then any of our native wild non-commercial plants in Massachusetts. And so we do our work for the benefit of all species, and really so that the people of the Commonwealth can continue to enjoy the natural and aesthetic qualities of the environment and going into future generations. One of the, I just mentioned it because I know a lot of people probably on here visit some of our properties, but one of the most important things we do as an agency is land protection. So we have a very rigorous land protection program where we focus on the most ecologically important or significant properties across the Commonwealth, like this one here. The Francis Crane Wildlife Management Area in Falmouth has one of the biggest and most important sandplain grasslands in the state, and is home to dozens of rare species, some of which that Mark Mello was talking about earlier. So to transition into talking about eagles, as I mentioned, they are one of the most well-loved species in Massachusetts and across the country, really, but that's not necessarily always been the case, and I'll get into that as much as I can in 15 minutes. The story of the bald eagle in North America is part gloom and doom and really part conservation success. But it is an important story to tell because it serves as a constant reminder of how humans' actions can have really terrible environmental consequences, but also that if we remain vigilant and take the appropriate actions, we can recover species from the brink of extinction. So the eagle story is part terrible and part great because they are one of our true conservation success stories. Few sites in nature people get more excited about than seeing a bald eagle, particularly in Massachusetts where there are relatively newly recovered species. People still get really excited and jazzed when they see an eagle. I do, and I've seen a lot of them. They were widely revered by Native Americans, chosen by our European settlers as the symbol of their new homeland, and they're really a national symbol of freedom. So they're one of those species that everyone loves and everyone likes to learn more about. So I'll get into a little bit of the natural history. You can only do so much in a little amount of time, but the scientific name, Haliaeetus leucocephalus, it's pretty basic. It means sea eagle with a white head in Greek. And that is the most common or conspicuous adult characteristic of this species, that bright white head with the yellow bill and yellow eye, and then the dark brown body with a white tail as well. You can notice here the sharp hooked beak that they use to tear flesh from their prey. Bald eagle is our largest raptor in Massachusetts. They can have a wingspan up to about 8 feet. And like is the case with many bird species, not all, the female are considerably larger than the males. And that's particularly the case with raptors, but a number of bird species, because they bear that additional burden of creating and laying eggs. The females can weigh up to about 14 pounds, while males will typically be in the 7 to 10 pound range. And in the picture here, you can notice that rounded tail, which is characteristic of them. And their head and neck is usually about equivalent to the length of their tail. You can also see the characteristic dark brown body with the white head and tail, and that's the adult characteristic. Their flight pattern is pretty discernible. Oftentimes people will see vultures, which have that dihedral pattern. Eagles take really deep, broad strokes when they're not soaring. So once you've seen one, you immediately can recognize that, that flight or wing beat pattern. Their age, they can live up over 20 years of age on average in the wild. Some have been known to live over 30 years, and I think the oldest known one up to this point was an eagle that lived to 38 years old. So they're a fairly long-lived species once they reach that adult stage. Their survival rates when they're younger can be much less. We do have golden eagles in Massachusetts as well, but they aren't quite as big as the bald eagle, and they're just more of a periodic visitor to the state. So to quickly go through the feather characteristics, so when they're first born, once they fledge the nest that first year, this is what they look like, just a large brown kind of nondescript bird. Their second year, they start to show some white feather patterns in areas where they won't have it as adults, so it's kind of interesting. They get into that third and fourth year, so going back and forth from third to fourth. This is the time period where they're commonly referred to as dirty birds. So they look kind of like an adult, but they have that pattern of brown on their head and tail that looks like they rolled around in the mud and got dirty. And then once they've reached that fifth year, they have they're full adults, they're sexually mature, they have the bright white head and tail, dark brown body with that bright yellow beak and yellow eye. Range of eagles in North America, they range pretty much throughout most of North America, from Alaska and some of the Canadian provinces, right down through parts of Central America. In Massachusetts, they are found statewide, but especially during migration in the spring and fall, they can be found pretty much anywhere in the state. Most people know that they're a bird that likes to be around water, and that's because of their main prey, which is fish. But they historically were tied to something like this, very large bodies of water with relatively undisturbed shorelines. As time has gone on and perhaps eagles have shown their adaptability, we're finding them more and more in areas where there's a lot of people and the shoreline isn't necessarily unbroken like you can see here. But this is what I would characterize as classic ideal bald eagle habitat. Their food habits, they are a fish eagle. Their primary source of food is fish. However, they will regularly prey and heavily prey on waterfowl, in particular in the wintertime when waterfowl get really concentrated and there's ice in other areas and maybe their access to fish is limited, they will really key in on waterfowl. They're also known to eat gulls, other birds, and in the wintertime, again, in particular, carrion can be, or dead animal carcasses can be pretty important, particularly like a deer getting out on the ice and they break their legs. You know, we've had instances in my district down in Plymouth where we had a deer out on the ice, and I counted, I think it was 9 different eagles that were utilizing that carcass. So talk a little bit about their breeding and nesting behavior. So eagle pairs will typically begin prepping their nests, you know, adding and rearranging sticks and adding soft nest bowl material anywhere from 1 to 3 months prior to mating. And this is really the time period when we're looking to the public for reports of adult eagles. So starting back in maybe early February through the end of this month, we really want to hear about where people are seeing adult, you know, full adult eagles, because generally they're going to be very close to or within their nesting territory. They're really active this time of year. You can see them carrying large sticks. So if anybody wants to report observations to us, you do see an eagle carrying a stick, just make a quick note of the location and the direction they were headed with the stick, because they're headed to their nest site. Nests are constructed in large trees at or near the water's edge, typically, or anywhere from right on the water's edge or overhanging the water to 80 to 100 feet inland, depending on the topography. And they'll go anywhere from 30 feet off the ground to as high as 120 feet or higher. And generally, the nest will be anywhere from 5 to 10 feet below the crown of the tree. And most of the thought behind that is they want a little bit of cover, some leaf cover or needle cover above them to help shade the nest and protect some, provide some cover from the elements for the young. These nests can get huge, so anywhere from 8 feet wide to 12 feet high, and they can weigh over 1,000 pounds. Some of the nests that we've climbed into are almost getting to the point where we can't climb into them anymore because we can get up to the base of the nest, and then we don't have anybody big enough or tall enough to be able to reach into the actual nest and handle the young. So they're pretty amazing. Eagles lay typically 2 to 3 eggs, but anywhere from 1 to 3. I've never seen 4 in any of the nests that we've dealt with. The eggs are pretty nondescript, and I'll get into that there. There's the nest, the eggs. You can see they're kind of a basic looking, chicken egg looking egg. Nothing too, particular about them. They're just a white coloration. The young will be born. We have some nests right now. We keep track of all our nests. They can be born anywhere from, mid-March through, the first couple of weeks of April, but we're about getting into the time period where we're going to start expecting some nests to hatch in a couple of weeks now. One of the interesting things about eagles is it says here, the early bird gets the fish. So the eggs don't hatch at the same time. One of the eggs will always hatch earlier than the other two. And that's, most likely an evolutionary adaptation so that one of the chicks will always be bigger and always be able to outcompete the other chicks in the nest for food, so that one chick is going to have the best chance of survival. We do have chicks that fledge, or nests that fledge 3 chicks regularly. So where they have good resources, they'll all make it. But if resources are tight or scarce, it's an adaptation so that one of the birds will survive to fledge. Oops. They grow astonishingly fast. So they're going to be brought 3 to 4 fish a day. They will reach 90%, or T90, which is the time to reach 90% of their adult weight, is only 42 days. So you're taking something that hatches out of a less than 3-inch long egg, and it's going to weigh 7 to 9 pounds in about 45 days. They will be fully feathered at 8 weeks. And around that time, around 8 weeks, is when they really will start to exercise their wing muscles and start to get prepared to take their first flight. So as I mentioned earlier, the story of the eagle is definitely two-sided. One that's it's got a terrible aspect to it, but then a really positive side of things. So this species was once considered a complete nuisance. It was thought mistakenly to be preying on livestock, when in reality it was eating livestock animals that had deceased for some other reason. All across the country, they were basically shot on site as a nuisance. In some states, including Alaska, there was a bounty on them. And in Alaska, between 1917 and 1940, well over 120,000 bald eagles were killed under that bounty program. So at the point in time when European settlers arrived, it was estimated that there was about 500,000 eagles in North America, with roughly 100,000 in the lower 48. And for a variety of reasons, just widespread killing, the bounties that were placed on them, and then DDT. So if anyone knows anything about DDT, it's a pesticide that was applied to agriculture, and nobody knew it at the time, but it ran off into the environment and through the trophic cascade, basically was accumulated by eagles and other birds of prey, and it caused an inability of them to produce enough calcium to produce good hard eggs. So the eggs would crack under the weight of the female. So it caused major declines in a lot of raptor species, but it's one of the key things that played a role in the decline of the bald eagle. By about the 1960s, there were less than 500 pairs of eagles in the lower 48, and they were essentially at the brink of extinction because of these multiple factors. And this was really the darkest time for the species in the species history. So that kind of leads into the recovery phase, and there's a number of laws and regulations that went into place, but there's also another significant event that I'll touch on briefly that led to their recovery. So I like to say Massachusetts leads the way in protection of eagles. So in 1907, the Massachusetts legislature passed a law that prohibited the hunting of eagles. It was a nice thing, it was a good thing, a nice first step, but there were a number of other things that had to fall into place in order to get the species back from the brink of extinction. The Migratory Bird Treaty Act was passed in 1918. That basically prohibited taking, killing, or possessing of migratory birds, parts, eggs, or products. However, birds of prey weren't added until 1962. So as great of a law as that is, it didn't do anything for eagles at the time. The Bald Eagle Protection Act was passed in 1940, and that basically made it illegal to kill, harass, or possess bald eagles, but Alaska, no surprise, was exempt there. And two laws that predated the actual Endangered Species Act were the Endangered Species Protection Act and the Endangered Species Conservation Act, and they really didn't do a whole heck of a lot when it came to protecting bald eagles, but it did authorize protection of important habitat for eagles and did afford them a little protection in terms of killing or possessing. The biggest thing, and I'm assuming most people on here are conservation minded, otherwise you wouldn't be on this conference. The biggest thing that happened was, if you're familiar with the book by Rachel Carlson called Silent Spring, that brought national attention to DDT and its impacts on bird populations and the plight of the bald eagle. And because of that book and the environmental movement that it started, the Environmental Defense Fund was established, and widespread grassroots efforts eventually led to pressure, and the EPA banned DDT in December of 1972. The next year, the Endangered Species Act was passed, and that basically afforded protection to a whole host of species, whether they were endangered or threatened. And up until not too many years ago, the bald eagle was protected under the Endangered Species Act. So to get into briefly some of the recovery efforts across the country, in Massachusetts, that involved what you're looking at here. This is the construction of what's called the Hacking Tower at Quabbin Reservoir. And there's a better picture of it here. And this is basically an artificial nesting structure that was created so that people could raise eagle chicks into adults in Massachusetts and release them into the environment. So a total of 41 chicks were released from 1982 through 88. Most of those were taken from wild nests in Michigan, Manitoba, and Nova Scotia. And then through the back where you see the ladder going up, a crew of people and volunteers would come up and feed the chicks, raise them to the point where then they could open the doors, they could go in and out, exercise their wings, and then fledge into Massachusetts with the hope that you were going to start to reestablish an eagle population again in Massachusetts. So I didn't mention earlier, I don't know why it keeps doing that, that our last wild nest in Massachusetts was, interestingly enough, on Cape Cod. It was at Snake Pond in Sandwich. So following the Hacking Tower efforts and the reintroduction efforts at Quabbin Reservoir, we had our first housekeeping nest. And for those of you who don't know, housekeeping is basically an adult pair has bonded and they go through all the motions of building a nest. They just don't lay eggs that first year. So we had our first nesting activity in 88, and then in 1989 we had 2 active nests that fledged 3 chicks. So over 80 years since the last nest in Massachusetts, we had eagles fledged again from wild nests. And since that time, we've had, I think, over 800 chicks fledged in Massachusetts. Looking at this graph here, you can see it trickled along for a while, and then now we're in a phase of almost exponential growth. It seems like every other week I get new reports of eagles or information on potential new eagle nests. So since the recovery plans were put into place and DDT and other harmful chemicals were banned, the number of eagles has met or exceeded many established goals under the Endangered Species Act and various recovery plans, to the point now where we have tens of thousands of breeding pairs between Canada and the United States. In Massachusetts in particular, we've gone from our first 2 nests back in 89 to now I know that we're well over 80 nests in the state, because this graph is a couple of years old. So we're in the monitoring phase now in Massachusetts. We continue to monitor the nests and follow up on any leads on nests. And it's an important part of ensuring that the bald eagle stays on a path to recovery. And we do this with numerous partners. Mass Audubon, a number of other organizations help us in monitoring the population and keeping track of banded eagles. So birders and wildlife photographers give us valuable information from our banded birds. So every year, except last year, we only did 1 nest because of COVID. But every year we typically get out and climb a good percentage of all of our active eagle nests in the state. And the reason for doing that is part of ongoing population monitoring. So we put a federal and a state band. The state band on the right, which is a color-coded and alphanumeric band that can be read by, again, birders, wildlife photographers, and that can give us information on the distribution and travel patterns of eagles that are born in Massachusetts. So to very briefly talk about nests in Southeastern Mass, we had our first confirmed nest in 1993 at Pocksha Pond in Middleborough. That nest or that pair has been active every year since then. We're not entirely sure if one of the adults could have passed and remated with a new adult, but it has been active. And now at this point in Southeastern Mass, we have 12 active nests, and I hesitate sometimes to put out numbers because I've already heard about a couple new potential nests. And then for the Cape Cod perspective here, this is the only nest that we banded last year. That's my technician, Jeff Breton, with the first bald eagle that was born and fledged on Cape Cod since 1905, the last nest in Sandwich at Snake Pond. So it was a pretty historic event. That's why we got approval from higher levels to go out and do the banding activity, because of the importance of this particular eagle in the recovery of the species, both nationally and in Massachusetts. So I mentioned, remaining vigilant for new threats. Back in the day when people were putting DDT out, and I know, it seems like every year people find out something that causes environmental harm that we didn't know of. So it's very important that we as a society keep vigilant for new threats that could exist. With the specific terms with regards to the eagle, some of the threats that we know of are just development of shoreline habitat. That's going to be a constant battle, because people and eagles both like to live right on beautiful shorelines, on water bodies and on the coast. Another thing that's important, and assuming there's a lot of birders and wildlife photographers that are present, is once we do have an active eagle nest, it's really important that the public enjoy them from a distance. And we've had to deal with a number of situations where people find out about a new nest, it's easily accessible, and then we have people camping like 30 feet away from the nest tree in lawn chairs with their cameras and stuff like that. And I just ask that the public use common sense and and get a good scope and observe the species from a distance, because they are susceptible to disturbance. And with that, I just want to wrap up. We do have 2 new nests that we've been reported to us on Cape Cod this year, one in Falmouth and one in Marston's Mills. So we're considering those active nests at this time. So we now have 3 active nests that we know of on the Cape. However, we've had reports from, Hinckley, Seymour, Sheep Pond area, Mashpee, Wakeby, Brewster, and a number of other sites across the Cape. So I'm certain that there's other nests out there that we don't know about. So we'd love to hear from the public on reports, or if you know where there's a nest, we can try and protect it. We usually put up signs and try and keep people away as best we can. And I'll wrap up here and thank everyone on this conference for the role that you each individually, whether in your personal life or through your professional careers, play in conservation in Massachusetts. I encourage you to reach out to me personally or our agency with any questions or issues you have with wildlife, because that's what we're here for. That's what I do as a public servant for the Commonwealth. And thank you all.
Excellent. Thank you, Jason. All right, so hopefully you can stick around for Q&A. Jason and I know Danielle are both still around. We've got a couple of questions so far. Feel free to keep typey more questions in the chat. So the first one, there was a question for Danielle a while back about ditch remediation and how that works, and then how long it would take to see an impact.
So for ditch remediation, what we use is salt marsh hay. So the high marsh plant, we kind of use that to strategically fill some of the ditches, not all the ditches, because we still want to have drainage within the marsh. So we determine which ditches are most important for drainage and which ones aren't. And the ones that aren't, we fill them with salt marsh hay. And so they say like monitoring for about 3 years. And then within that 3 years, you kind of treat the ditch as needed. And over time, you're able to kind of see like the ditch material, like what's within the ditch, resembling more of like that salt marsh peat. So I would say maybe about 3 years or so, you can actually kind of see like a big difference of how the ditches are kind of filling naturally. And it just basically accelerates a natural process of ditch filling, especially using the natural material.
While we have you and you're on the subject, I don't see a question about it, but there's another technique that's been in the news called runneling. Maybe you could address that one as well.
Yeah, so runneling, so I know there's sometimes like questions about like what's the difference between runneling and ditching. So runneling is shallow, because you can notice with ditches that you can see ditches that are meters deep and meters wide, versus runneling like maximum maybe about a foot deep or a foot wide, and oftentimes it's even less. So with runnels, you can actually potentially even like create a runnel using like the heel of your foot, or oftentimes these are just dug using shovels. And the purpose of runnels is to drain water impoundments that's resting on the marsh. Because with sea level rise, what we're seeing is that the marsh isn't draining as well, and it's not draining during low tide. So vegetation is being flooded for longer periods of time, and we're seeing our vegetation under higher flood stress. So, and we're seeing these water impoundments starting within the marsh, becoming bigger and bigger and bigger, which is leading to the marsh potentially eventually becoming a mud flat. So to prevent that, we use these runnels or these shallow channels to drain those water impoundments and to alleviate some of that flooding stress that the vegetation is under.
Very good. There's a question for Jason. He's answering it in the chat, but maybe you can also address it for the group. Sometimes what happens in these things is some people don't have access to chat for mysterious Teams reasons we don't understand. I don't know if that's behind some of the dearth of questions, but just in case there are people who don't have access to chat, we'll just, if you could just talk about where people can report, not bald eagle sightings, but specifically bald eagle nest sightings.
Yeah, so you can report them a number of ways. I mean, you can call our office, any one of our offices, district offices across the state, but at Southeast, you can just call our office. And that's the numbers available on the website. And then emailing me directly or our ornithologist, Drew Vitz, in our Westboro office, you can email him directly. So my email is jason.zimmer@mass.gov.
Right, and that'll be in the abstract packet as well. So I know there's a nest on Cape Cod that's a carefully guarded secret in terms of where it is. In some circles, it's a poorly kept secret, but have there been any whispers of any others? I know you won't tell us where, and you shouldn't. But is it possibly more than one nest on the Cape? I would imagine. Yes,
and we're certain that there's other nests out there that have been active for a few years that we haven't just been able to nail down yet. Mashpee, Wakeby, there's got to be something close by there. Right. But we do have 2 new reports this year, the one in Falmouth and then one down in Marston's Mills, and they both came with photographic evidence of eagles in the nest and eagles actually mating in the nest. So if they're not kicked out by osprey that show up, if it was an osprey nest, which happens sometimes, we should have at least 2 more definite active nests this year on the Cape.
Wow. All right. You heard it here first, folks. So a couple of people have raised their hand, which makes me wonder if they don't have chat. So there aren't that many people. Why don't we say, Maureen, if you could unmute yourself and ask your question if you had one. Unless that was an accidental hand raising, which also happens. You never know. These are weird times. Meetings are very strange now, let's face it. I actually got some laundry folded. Found that I could do that and pay attention to talks at the same time. We're all learning new things. All right, how about Pat Evans? Is that an intentional hand up? Did you want to unmute yourself and ask a question? These may be accidental hands. It happens.
I just wanted to
oh. I hear muffled murmurs.
Get there very soon. Thank you.
Anybody catch that question? Yeah, that was pretty muffled. Sorry. Well, maybe next year we'll be back at 4 C's like usual, seeing people in person. Oh, is there anything?
I just wanted to thank you.
Okay, go ahead.
Thank you for a wonderful morning. You gave out so much information. Your guests were so intelligent, but at the same time they were talking to the average person and bringing so much information in just ordinary language, so that it made me feel very comfortable listening to everyone.
Well, thank you. That was not a planted comment. Thank you for the Thank you for the compliment. Glad you enjoyed it. We did have a question from Lisa Meeks asking if anything preys on the young. I know the answer to this, and you can find videos of it. Is there anything that preys on the young eagles while they're still in the nest?
Yes, so you can have, there's a number of things that can actually prey on them. You could have, and we do have evidence of raccoons that'll go up and get into the nest, but the most common ones are going to be avian. So it's either going to be great horned owls, crows are one of the big ones that'll come in and take them when they're young. And you can have other raptors that'll come in and take them in addition to great horned owls.
Interesting. I knew about great horned owls, and there are videos out there of both great horned owls and eagle owls in Europe and Asia. It seems unbelievable, but they have the advantage at night, and they can go into a nest and take an almost fully grown osprey chick, or a fully grown or almost fully grown eagle chick, which is amazing. Okay, I think that's it for questions. So First, I'd like to thank my colleagues, Joel Wagner and Jeanette Kerr, who are kind of behind the scenes and who both really did most of the work organizing this year's conference. Also, Ian Ives at our Long Pasture Sanctuary. I want to thank all of our speakers, Lisa, Mark, and Danielle, and Jason, and Charlie, and who else?
Megan.
Oh, Megan, of course. She's not here. It's okay. Thanks to Megan. That was really cool stuff with the sharks. I'd like to thank our sponsors again, the Birdwatchers General Store, and also, no more questions, the Cooperative Bank of Cape Cod. And so on behalf of the Mass Audubon Cape Cod, and our director, Melissa Lowe, thanks to all of you for attending. And hopefully we'll see you next year in real life.